When scientists compared Black American DNA to British DNA, the results did not just reveal ancestry, they revealed history. Specifically, the history that was deliberately erased. The genetics showed which African ethnic groups Black Americans descend from, how much British DNA is in the average Black American genome, and exactly how it got there. The data was precise. It was undeniable. And for millions of people, it was the first time science gave a name to something their families had carried for 400 years. Before we dive in, comment where you are watching from and click subscribe. Let's start with what geneticists actually do when they compare two populations. Imagine you have a book written in a language made of 4 letters, A, T, C, and G, and that book is 3 billion letters long. That is your genome. Every human being on Earth has one. And at the vast majority of positions in that book, more than 99%, every human being has the same letter. The differences between any two people lie in the remaining fraction of a percent. But in that fraction, across 3 billion base pairs, there is an enormous amount of information. Scientists call the specific positions where people tend to differ SNPs, single nucleotide polymorphisms. And by looking at hundreds of thousands of these positions simultaneously, geneticists can do something remarkable. They can calculate with statistical precision where a person's ancestors came from, which populations they descended from, and crucially, what percentage of their genome traces back to each of those populations. Think of it like reading the rings inside a tree trunk. Each ring tells you about a specific year, how much rain there was, how warm the summer was, whether the tree was stressed or thriving. A geneticist looking at your genome is doing something similar. Each segment of your DNA that matches a particular population is a ring, a chapter, a signature left by an ancestor who came from that place. And when you line up the rings of millions of people and compare them, the patterns that emerge are not just scientifically interesting. They are historically explosive. When they applied these methods to tens of thousands of Black Americans and compared the results to British and other European populations, the data told a story that reached back 4 centuries. And it was more complicated, more painful, and more surprising than almost anyone expected. Here's the first finding, and it is the one that reshapes everything else that follows. The average Black American genome is not entirely African. According to the landmark 2015 study published in the American Journal of Human Genetics by Dr. Kajiya Bryce and colleagues at 23andMe, which was the most comprehensive genetic analysis of American ancestry ever conducted at that point, and drew on the genetic data of over 160,000 customers, the average Black American genome is approximately 73.2% African, 24% European, and 0.8% Native American. The European fraction, nearly a quarter of the entire genome, is what the comparison to British DNA unlocks. Because when you look at where that European ancestry traces back to, you are not looking at random European admixture from across the continent. You are looking overwhelmingly at one specific corner of the British Isles. According to data from Ancestry.com and multiple independent genetic studies, the specific European regions most commonly appearing in Black American genomes correspond to the populations that dominated Southern white colonial and antebellum society: English, Scottish, and Irish. Not Spanish, not French, not German, British. The British Isles. The very same populations that sailed to North America, built the colonial economy on the backs of enslaved labor, and whose genetic signatures were then incorporated through violence into the genomes of the people they enslaved. That word violence matters here, because when you look at how the British DNA entered Black American genomes, the genetics do not just tell you that mixing happened, they tell you the direction of that mixing. And that direction is specific, documented, and damning. In August 2023, Harvard Medical School published a landmark study analyzing the DNA of 27 enslaved and free Black individuals who lived and died at Catoctin Furnace, an iron forge in Maryland between 1774 and 1850. Researchers compared their genetic data to the 9 million person database of 23andMe, identifying over 41,000 living relatives. What they found about the European ancestry in those genomes was precise in a way that purely historical records never could be. The European ancestry in those individuals traced primarily to Ireland and Great Britain, and it arrived overwhelmingly on the paternal side, not the maternal side, the father's line. What that means genetically is not ambiguous. European men were contributing DNA to the children of enslaved black women, not the reverse. The pattern is the genetic fingerprint of systematic sexual exploitation, of enslaved women being raped by their enslavers, the majority of whom were of British or Irish descent. A separate study by 23andMe, published in August 2023, analyzing DNA from individuals connected to early enslaved and free African Americans, found the same pattern. The European ancestry in those early genomes traced mainly to Britain and Ireland and was overwhelmingly on the paternal side. The study stated directly that this reflected the documented historical phenomenon of enslaved women being raped by their white enslavers and others in positions of power. So when scientists compared Black American DNA to British DNA, what they found was not a genetic curiosity. They found a forensic record of 4 centuries of American history written in chromosomes. If you are getting information from this channel that you cannot find anywhere else, take 2 seconds and subscribe. We cover the science that goes deeper than the surface. Now let us go deeper. Because here is where the comparison between Black American DNA and British DNA produces its most shocking finding, and it runs in the opposite direction from what most people assume. We have been talking about the British DNA inside Black American genomes. But what about Black American DNA that has made its way into British genomes? Researchers, funded by the Wellcome Trust and by the European Union funded Eurocourse project, made a discovery that quietly upended what the British thought they knew about their own genetic history. Published in the European Journal of Human Genetics, the study found the first documented evidence of African genetic ancestry in the indigenous British gene pool, ancestry that dates back at least 250 years before the mass immigration of the mid-20th century. The researchers found an African Y chromosome passed from father to son through the direct male line, embedded in what had been considered the indigenous British population. Africans had been arriving in Britain for far longer than most British people realize. Some of the soldiers who served the Roman Empire in Britain nearly 1,800 years ago were African. There are historical suggestions that Vikings may have brought captured Africans to Britain in the 9th century, and by the 16th century, African servants, musicians, and enslaved individuals were present in British cities. Their DNA, in some cases, made it into the British gene pool and has been there ever since. The flow of DNA between Black Americans and the British, in other words, was never a one-way door. It never has been. Now let's talk about the African side of this comparison, because this is where the science produces its most emotionally powerful findings. When researchers compared the African component of Black American DNA to populations across the African continent, they were able to answer a question that had haunted African American families for generations. Where in Africa did we come from? The landmark study led by Dr. Sarah Tishkoff of the University of Pennsylvania and Dr. Carlos Bustamante of Cornell, published in the Proceedings of the National Academy of Sciences, analyzed the DNA of 365 African Americans alongside 203 West Africans from 12 countries and 400 Europeans from 42 countries. What they found about the African roots of Black Americans was granular in a way that no historical record could ever be. The West African component of African American DNA most closely matched the genetic profile of the non-Bantu Niger Kordofanian-speaking populations, specifically the Igbo and Yoruba from Nigeria, and the Brong from Ghana, the Igbo and the Yoruba. 2 of the 10 most frequently documented ethnicities in slave trade records, confirmed now not just by historical documents, but by the genetic data of living people. The genetics confirmed what the historical documents had long suggested and made it personal in a way that no archive ever could. Additional research from family tree DNA, 23andMe, and MyHeritage identified further specific genetic connections linking Black Americans to the Wolof and Mandinka of Senegal and Gambia, the Akan of Ghana, the Mende of Sierra Leone, and the Fula people of the broader Sahel region. These were not abstract continental origins. These were specific named peoples with specific named cultures, specific languages, specific kingdoms, all traceable through the biological record even when every other record was destroyed. A random Black American born in 1960, according to a USC and Stanford University study published in the journal Genetics in 2023, is descended on average from 314 African and 51 European ancestors, dating all the way back to 1619. Think about that number. People who were captured, marched to the coast, loaded onto ships, and transported across the Atlantic. People who survived the Middle Passage. People whose names were deliberately erased from the historical record, but whose DNA, whose actual biological information survived, reproduced, and persisted through 14 generations of American history to the present day. And here's the comparison that the title of this video promises. When scientists lay the average Black American genome next to the average British genome and compare them, what do they see? On the surface, the two populations appear to share European DNA, but the character of that shared DNA tells completely different stories about how it got there. The British genome, according to the landmark 2015 Nature study led by Dr. Peter Donnelly of the Wellcome Trust Center for Human Genetics in Oxford, the most comprehensive genetic analysis of British ancestry ever conducted, drawing on 2,039 carefully chosen individuals, is itself a layered archaeological record. Modern British DNA contains the signatures of at least 4 major ancient migrations. The original post-Ice Age hunter-gatherers who settled Britain around 12,000 years ago, a wave of Neolithic farmers who arrived from Anatolia in modern Turkey, roughly 6,000 years ago, the Bronze Age Bell Beaker migration that arrived around 2,400 BCE, and according to ancient DNA research published in Nature, replaced approximately 90% of the existing British gene pool within a few hundred years, and the later Anglo-Saxon migrations of the 5th and 6th centuries CE, which contributed between 10 and 40% of ancestry to people living in eastern, central, and southern England today. A 2026 study published in Nature further refined this picture, finding that the Bronze Age migrants who reshaped Britain arrived not from Iberia as older theories suggested, but from the river delta communities of the Netherlands and Belgium, people who carried a blend of steppe ancestry from the Russian steppes mixed with older hunter-gatherer DNA from the wetlands of northwestern Europe. Even the relatively recent Viking Age left its mark. The population of Orkney in northern Scotland carries approximately 25% Norwegian ancestry, a remnant of Viking settlement that is still visible in the genomes of people living there today. The British genome, in other words, is not an ancient, unchanged European lineage. It is the product of migration after migration after migration, populations replacing and mixing with each other across thousands of years. People came from the Russian steppe, from Anatolia, from the river delta communities of what is now the Netherlands and Belgium, and from Scandinavia. The British genome is, at its core, a story of constant movement, of replacement and admixture stretching back to the end of the last ice age. What we think of as British DNA is, genetically speaking, one of the most thoroughly remixed data sets in all of European history. Now look at the Black American genome through the same lens. It contains the signatures of West African populations, the Yoruba, the Igbo, the Akan, the Mandinka, the Wolof, the Fula, peoples from across the arc of the West African coast. It contains British and Irish ancestry, mostly forced, mostly paternal. It contains a small but measurable fraction of Native American DNA, reflecting contact between enslaved Black people and indigenous populations, particularly in the Southeast. And because all of these admixture events happen within a specific and well-documented historical window, primarily between 1619 and 1865, scientists can date them with unusual precision. The 23andMe researchers found that the mixing of African and European DNA in Black Americans occurred almost entirely before the Civil War, mostly concentrated in the South during the era of chattel slavery. What that means is this: while the British genome is layered across millennia, mixing events separated by thousands of years, the Black American genome compresses a comparable degree of genetic complexity into a single, brutal 400-year window. The same diversity of origins, a fraction of the time, and every single admixture event on the European side carries the specific historical context of enslavement, exploitation, and violence. There is one more dimension to this comparison that the data reveals, and it may be the most counterintuitive finding of all, because the common assumption is that of these two populations, the British are the genetically stable, continuous reference point, and Black Americans are the admixed, complex population. The data challenges that in both directions. Black Americans, despite the enormous violence of their history, show lower levels of what geneticists call runs of homozygosity, long stretches of identical DNA on both copies of a chromosome. Some highly isolated British subpopulations, like those found in Orkney, trace 25% of their DNA to Norwegian ancestors, and centuries of geographic isolation have produced distinct genetic signatures. Genetic diversity, in other words, is not a measure of trauma. It is a measure of how many different lineages have contributed to a genome. And Black American genomes, because they draw from both West African populations and European populations, contain some of the highest levels of genetic diversity of any population in the United States. What was forced upon a people as an act of destruction also created biologically one of the most genetically rich population histories in the Americas. That is not a justification of what happened. It is simply what the science shows. The genome does not lie. It does not forget. It does not take sides. It only records. What does all of this mean when you put it together? It means that the comparison between Black American DNA and British DNA is not simply an ancestry exercise. It is a forensic accounting of one of the most consequential historical relationships in the modern world. The British colonized North America. They built an economic system dependent on enslaved labor. They transported, bought, and sold African human beings, primarily West Africans, primarily Yoruba and Igbo, and Akan and Wolof and Mandinka. They exploited enslaved women. They produced children, and those children and their children and their children's children for 14 generations carried the genetic evidence of all of that forward in their bodies. The study from USC and Stanford estimated 314 African and 51 European ancestors for a single Black American born in the early 1960s. The 51 European ancestors were, in most cases, not there by choice. The genetics make that clear. The paternal side concentration of British and Irish DNA in the earliest documented Black American genomes is not ambiguous. It is one of the most consistently documented findings across multiple independent studies, from Harvard Medical School to 23andMe to the University of Pennsylvania. But the other side of the accounting is equally important. Those 314 African ancestors, each one of them a person, each one of them the carrier of a genetic lineage that stretches back thousands of years into West African history, into the Yoruba kingdoms of Nigeria, into the Akan states of Ghana, into the Senegambian empires of Mali and Senegal, into civilizations that had art, architecture, governance, spirituality, and culture for millennia before the first slave ship crossed the Atlantic. The genetics can trace those lineages. They can point to the specific ethnic groups, the specific regions, the specific river valleys from which those people were taken. They can give back, in the language of DNA, what 4 centuries of forced amnesia tried to permanently delete. That ancestry, all of it, survived. The records were burned, the names were changed, the languages were beaten out of people across generations, but the DNA persisted. It copied itself faithfully in every cell of every child born into bondage. It passed through the Middle Passage. It passed through Reconstruction. It passed through Jim Crow and the Great Migration and every generation since. And now, for the first time in human history, the tools exist to read it, to decode it, to restore, at least in biological terms, a portion of what was stolen. That is what scientists found when they compared Black American DNA to British DNA. Not just a genetic comparison, a reckoning. If this video gave you something to think about, share it. Drop a comment below, and if you want to keep going, hit subscribe. Because next time, we are taking this somewhere even more unexpected. Scientists recently compared the ancient DNA inside West African genomes to populations that predate any known civilization. And what they found living inside those genes rewrites the timeline of the entire human species. That one is coming soon. You do not want to miss it.